So thank you for the invitation. My name is Paola Crepaldi, and I work at the Department of Agricultural and Environmental Science at the University of Milan. I am a geneticist and I work on biodiversity in livestock species, especially in small ruminants, and also in companion and especially in dogs. Today I will talk about dog biodiversity and how we can move from describing it to managing it using genomic tools. This is a joint presentation. I will cover the first part while Dr. A.Bionda will follow. We will show how genomics combined with traditional breeding can support better population management, sometimes already well established in livestock, but increasingly important in dogs, which are both managed populations and companions. So dog biodiversity comes from a long evolutionary journey that started with a wolf domestication. All modern dogs share this common ancestor, but thousands of years of human selection and adaptation have shaped the diversity we see today. The first major change in domestication was the loss of camouflage, typical of the wild species. Other traits such as white patches, floppy ears, shorter muzzles, and smaller size are known as domestication syndrome, linked to the selection for tameness. This is well illustrated from the Belayev's silver fox experiment, where selecting only for docility led to all these typical domestic traits. From domestication, we move to the formation of early functional types that we could call protobreeds. Across time and cultures, dogs were selected for specific roles such as hunting, guarding, and companionship. So even in ancient time, dogs were not just companion, they were shaped for function. The modern concept of breeds emerged in the 18th century with pioneers like Robert Beckwell. He introduced selection based on an ideal type and emphasized the importance of recording pedigree. The first stud book appears in horses and later spread to other species. And this period saw the rise of breeder association, and with breed formation, population became more defined and closed. Standard described the ideal animal, and stud book recorded ancestry. This concept was soon applied to dogs through breed registries, standard, kennel clubs, and pedigree. From this point on, belonging to a breed depends on ancestry and on an appearance described in the standard. So breeding became more structured with controlled mating and selection of desired traits, often aesthetic. Darwin already described this process in the variation of animals and plants under domestication when he devoted an entire chapter to dogs. And he noted the rapid spread of different breeds, but also pointed out the risk of inbreeding and the loss of older population. This process created the remarkable diversity we see today, around 400 breeds, with extreme variation in size, morphology, function, and behavior. However, it has also reshaped the genetic structure of dog population, and this is a clear example of human-driven diversification. And this human-driven diversification is still ongoing. Some breeds have become global companion, like the golden retriever, while aesthetic selection has led to more extreme types like Neapolitan Mastiff or crossbreeding like Labradoodle. Urban environment tend to favor small dogs, while in rural areas there are still local working breeds, and in this case, you can see the Brahman sheepdog, originally local, but now a cosmopolitan breed, while the Brahman hounds remain typically local. However, close population come with a cost. Individuals become more related, and this reduces genetic diversity and increases inbreeding, depression, and the risk of hereditary diseases. So the question is, can we manage this? The answer is yes, and genomics can play an important role. Genomics provides powerful tools to better understand and manage genetic diversity at both population and individual levels. Our ability to understand genetic diversity has evolved over time. We started with Mendelian genetics, looking at how single traits are inherited. Then population genetics helped us understand how variation change across generation in a population. A major step forward came with genomics. The dog genome was sequenced in 2005, making it possible to study genetic variation at DNA level. And now with epigenomics, we can see how gene activity is regulated and influenced by environmental stressors such as nutrition, living condition, and so on. So today we have much more powerful tool to understand and manage genetic diversity. If we take a step aside and look at livestock, genomics is already part of routine breeding. This is because livestock breeding has long been based on population level approaches, with the systematic collection of phenotypic data from nose to tail, and more recently, genomic data. This allows reliable identification of animal parentage, of animals, parentage verification, genomic selection through the estimation of breeding value for complex and simple traits that are goal of selection, defined by breeder association. This and also genomics support the monitor of inbreeding and the management of genetic diversity. Moreover, it allows the identification of traits of interest and the identification of disease carriers of genetic disease, such as breed traceability. At the same time, it can provide information on how populations adapt to different environments. And at a global levels, database like the FAO database of animal diversity, that is, show that the animal genetic resources have a clear pattern. In cattle, for example, we have many local breeds around the world, each adapted to different conditions, and a smaller number of widely spread breed that are registered regional or international transboundary breeds, which tend to be more genetically uniform. This database help us monitor diversity and support conservation. It is important to note that at first, breeder and their association were quite skeptical about the application of livestock of genomics tool. There was initial resistance, as often happens when new technology are introduced into breeding system, but over time their value became clear. Today, genomics improve both breeding decision and the long-term management of population and allows to evaluate the genetic progress of population with respect of the goals fixed by the breeder association. But when we look at dog, the situation is quite different. So what can genomics do in dogs? So surely you know that genomics allow us to reconstruct the relationship among breeds, as you can see in this tree, connecting different functional group of dogs. Instead of relying only on records, we can analyze DNA directly And this reveals cluster and connection, and in some cases also unexpected historical links. For example, in one of our studies on Italian dog breeds, when compared with other breeds, we found that the Italian Shepherd dog breeds show the close relationship with the New World dog clade, likely reflecting a shared ancestry linked to the German Shepherd dog. Genomic allow us also to measure inbreeding directly from DNA, unlike pedigree-based estimates which provide an average expectation. Genomic inbreeding reflect the actual homozygous regions in the genome of each individual. This means we can estimate how much inbreeding is present in a dog or in a breed, as shown in the figure on the left, by the total length of the bar from the most inbred at the top and the least at the bottom. We can also see where these regions are in the genome and which genes are inside this region, and whether the inbreeding is recent, shown by the last red and green segment, or more ancient, shown by the initial pink segment. This gives us insight into how well a population is currently managed. So we can directly look at these indexes. At the same time, we can estimate effective population size, the figure on the right, that is the number of individuals effectively contributing to the genetic diversity of the population. And this also allows to reconstruct population history, for example, by identifying bottlenecks, which in many dog breeds often coincide with the moment of breed recognition. Together, these measures describe both current diversity and population history and offer information about the management of each breed. We can analyze also the genomic background at both the population or at individual levels. In this figure, each bar represents, in different colors, represent a breed composed of multiple lines corresponding to individual dogs. And the different colors indicate the contribution of different genomic backgrounds. Starting from the top left, some population cluster very clearly, showing high within breed uniformity, as in the Bracco breed in this case, shown here in a bright pink. In the top right, other populations are more complex, reflecting a mixture of ancestry linked to their selection history, and here represented by a mix of colors. In the bottom right, other population in the bottom right, some breeds are genomically very similar despite being classified as distinct. For example, the two Italian hound breeds. And finally, in the bottom left, we can see individual differences. Some dogs are fully assigned to their breed, show entirely the entire line in red, while others show shared ancestry with different populations indicated by multiple colors. These analyses are very powerful, and today many commercial tests provide this type of information. But what really makes the differences is the reference database. Its size and the number of breeds included are crucial for a reliable interpretation. So these results are useful, but they always need to be interpreted with care. So if you check your dog with two different commercial panels, the results could be slightly different. Genomics help link genotype to phenotype. So help link genetic variant to traits such as size, morphology, and also behavior. In the plot shown in this slide, chromosomes are represented in alternative black and gray colors, and the red peak indicates genomic region associated with the traits. For simple traits, first do, we usually see a single strong peak reflecting the effect of one major gene, as in a coat varnishing and fur length. In contrast, complex trait shown below, age and longevity, display multiple peaks indicated that several genomic regions are involved. So simple traits are easier to study, while complex traits require larger data sets and more detailed phenotyping. Genomic also allows us to identify disease-causing variant and develop genetic tests. Often this process starts from the field, from reports by owner, breeder, or veterinarians, and can lead to the identification of the causative mutation and its mode of inheritance, and eventually to the development of a genetic test. These support diagnosis, carrier detection, and more informed breeding decision. As shown in this slide, for example, a study on Sirnico dogs in the USA started from cases of retinal degeneration associated with neurological symptoms such as tremors, led to an international collaboration which allowed the identification of the gene and the mutation involved. The condition was named Sirnico ocular neurological syndrome and is caused by a one base pair deletion in the AMPD2 gene with a recessive mode of inheritance. Also for disease, results need to be interpreted carefully. Not all variants are known. Some disease has a long, a complex gene basis. Not all carriers will develop the disease, and the same condition in different breeds may be caused by different genes or mutation. Overall, genomics provide powerful tool to move from unexplained cases to practical solution, supporting both disease control and more informed and sustainable breeding. So, to conclude this first part, these tools already exist and are already being used. We can study diversity, understand the relationship, and manage genetic risk. So the question is, no longer do we have the tool. The real question is, how are we using them, or what are we still missing? And this is exactly what my colleague will address next. And so I let the floor to Arianna.
Thank you. I think that you should remove the sharing. Maybe I'm not sure that I can. Okay. Okay. Okay, can you see the screen?
Yes, we can see it fine.
Thank you. So let's start. Thank you for introducing me. And let's start from this question. How are we using these tools that are available also for dogs? To understand this, it helps to look at the system where these tools are already fully integrated, that is livestock. As we have already seen in livestock, genomics is not a standalone tool, but it's part of a broader breeding framework built at a population level. Moreover, phenotypic data are collected systematically and in a standardized way, so selection is based on measurable traits recorded over time and analyzed in relation to the whole population. Therefore, pedigree, phenotypes, and genomics are used together within a shared selection objectives and coordinated breeding programs. In dogs, the logic has historically been different. Breeding revolves around a breed standard, and phenotypic evaluation is often based on morphology, show results, or working tests. So phenotype matters here too, of course, but the way it is recorded is often less continuous and less consistent, especially for health and other complex traits, despite the efforts that I really appreciate and that have been highlighted today and yesterday. Pedigrees are also central, but they are often used mainly to define descent and breeding belonging, that rather than be a part of a full integrated selection system. And genomic tools are available and get used, think parentage, ancestry, or specific disease tests. But results usually remain private instead of being evaluated within a coordinated population-level strategy. And this leads to the key issue. The problem is not the lack of data or tools, because in dogs we have a great deal of information, but the challenge is that these data are often not connected. So they end up being used separately for individual dogs or decisions instead of powering a population-level management. And this is exactly where the opportunity lies. Because to manage population, the first step for us should be to monitor them. And this applies to dogs as well, because dog breeds represent a form of biodiversity, not only biological, but also cultural, often linked to human activities and local traditions. At the moment, most data are collected at the national level, often through kennel clubs. And this provides valuable information, obviously, but still only part of the picture. International systems such as the early mentioned FAO, that is, are designed exactly for this purpose, to extend this perspective across countries, bringing together information to follow population dynamics. Dogs are also included in that. However, as you can see here, at present, only a limited number of countries actually use it for dogs, and often report only a subset of their national breeds. So the framework exists, but its implementation is still incomplete and uneven, and does not yet provide a reliable global overview. And this matters because monitoring is not just about counting breeds, it allows us to follow trends, detect declining population, and assess risk status. If we want to properly protect the biodiversity and avoid losing population without even realizing it, we need to monitor dog population as a whole, not just the separate national registries. Monitoring is essential, but it is only the first step. To actively manage population, we need systems that can integrate different types of data, pedigree, genomics, and phenotypic information. We also need to analyze population over time, tracking changes in inbreeding, genetic diversity, and allele frequency. This requires infrastructure that can store data over the long term, analyze them, and return results in a form that is usable for breeders. And most importantly, such systems must act as a bridge between data and decisions. So the goal is not simply to collect the data, but to actually use them to guide breeding at the population level. One of the clearest applications of population-level genomics is the management of inbreeding. This allows us to evaluate individual animals within the context of the population, while also providing a global view of the genetic status of the breed. Pedigree-based tools are already used in many kennel clubs to guide mating choices. Genomics adds an important advantage because it measures the real sharing of DNA rather than just the expected one. In livestock, this is already standard practice, with breeding associations suggesting mating plans that balance selection goals and inbreeding. In dogs, this approach might also be applied. And when genetic diversity becomes more limited, managing mating alone may not be sufficient. In these cases, it might be necessary to broaden the genetic base of the population. Here, genomics can allow us to identify individuals beyond the pedigree that are both phenotypically and genomically consistent with the breed, for example, by analyzing their genomic background, as discussed earlier. These individuals can be introduced also in a controlled way, for example, through special registry sections. In addition, similar genomics approach can also be applied in structured crossbreeding or outbreeding programs, which are currently being explored in some countries for specific breeds, as also highlighted in some of the presentation yesterday. In livestock, such programs are well established and use genetic information to guide breed choice and to monitor how different genetic contributions are maintained across generations. As you know, applying this approach is not straightforward because the balance is delicate. Improving genetic diversity or specific health traits while preserving the defining characteristic of the breed. When combined with phenotypical, functional, and clinical evaluation, Genomic can support this process by tracking overall genetic diversity and known disease-associated variants, and by helping to characterize both the overall genetic background of each individual and how specific genomic regions are inherited, allowing thus a more informed selection of animals to be carried forward. Health is another area where a population approach makes a major difference. Everything starts with reporting, because without cases, there is nothing to investigate. From there, what really matters is having the right data. This means accurate phenotyping and a sufficient number of samples to detect consistent patterns. In this context, storing biological materials, for example by establishing an international biobank, is essential because it allows us to go back across generations and reconstruct how a condition originated and spread. And when these elements are in place, genomic analysis can identify causative variants and translate them into genetic tests. And once a variant has been discovered, the perspective shifts to the population. We can estimate how frequent it is, how it is distributed across families, and how it changes over time. This makes it possible to guide breeding decisions, not only to avoid producing affecting dogs, but also to progressively reduce the frequency of the variant in the population over generations. So health improvement becomes a continuous process rather than just a reaction to individual cases. Genomics also supports selection and evolution of breed standards. On one side, it allows us to identify genetic variants associated with traits of interest for breeders, from morphology and coat color to more complex traits such as behavior and working abilities. This supports a better understanding of the genetic basis of traits and allows more informed selection, particularly when phenotypic data are collected accurately and ideally in an harmonized way. At the same time, this data can support discussion on selection goals at the population scale, not to replace breeder expertise, but by providing a clearer and shared picture of the breed, which can help guide choices in dialogue with breeders. These decisions are closely linked to breed standards, which both describe the current population and guide its future development. Population-level genomic data help connect these two aspects. For example, when a trait is already present in the population but not included in the standard, it can be considered for reintroduction, as seen for the black coat color in the Volpini Italiano, or, to make a zootechnical example, for the red spotted in Holstein cattle. Similarly, this approach can be used when standards are modified to reduce extreme morphology traits and improve animal welfare. In these cases, genomics can support these changes by clarifying the genetic basis of the trait involved and whether the animals with the desired phenotype are otherwise consistent with the breed. This makes it possible to develop a more evidence-based and shared selection strategies and standards. And an important point to keep in mind is also that populations are not static. They continuously change due to selection, changes in the function of the animals, and also new knowledge and societal expectations. This means that population management cannot be a one-time action. It has to be continuous. We need to track how populations evolve over time, monitoring inbreeding trends, allele frequency related to traits and diseases, and changes in the genetic backgrounds, such as introgression or increased homogenizations. This also includes preserving genomic data in the raw form so that it can be reanalyzed in the future as new tools and methods become available, and collecting accurate and consistent phenotypic data over time. This approach makes it possible to evaluate the genetic progress of the population across years and generations, incorporate new phenotypes as they become relevant, and support shared long-term selection goals. And obviously, the longer we collect data, the more powerful they become. So population management means learning from the past while guiding the future. So to summarize, I want to say that dog breeds represent a remarkable and valuable form of biodiversity that deserve to be understood and preserved. At the same time, dog breeding already has strong foundations, pedigree, selection practices, and a deep knowledge of the breeds. Genomics does not replace this, but complements it and can reinforce it. Today, the main limitation is less about the tools themselves and more about how they are used. The key shift is indeed moving from individual decisions to decisions that take into account population as a whole. This means integrating knowledge, combining the experience of breeders with scientific data, ensuring that data are collected and maintained over time, and strengthening the collaboration between breeders, associations, veterinarians, researchers, and other stakeholders. And this is what allows us to move from improving individual animals to improving entire populations. So I want to thank all those that collaborated with us in our projects, as well as the owners, veterinarians, and the breeders that provided us data and information. And obviously, I want to thank you for your attention.